section is very much about the plasma membrane. It's about transport across it. So as many times as I've mentioned this structure, it's important to remember. So it is primarily a phospholipid bilayer. And it has proteins that go either through it or on the side. And as I learned just a few weeks ago, there's some that just act as little rafts on top. So there's many ways that these proteins can be aligned here. What are they called? Peripheral and integral? Oh yeah.
Oh, integral protein.
Okay.
And the peripheral protein.
So what is this one here in the front? What is that one? Extra.
I don't know. Something.
Yeah. Alright. What is this one that goes all the way through?
Protein.
What kind of protein is it?
Embedded.
The word. Okay, embedded. What's the other word for embedded?
Integral.
Integral. What is one that's just on the surface here? Peripheral. Because that's another word for periphery. Periphery is on the side of things. So here is that membrane. And remember that it's fluid. It's constantly moving. So it's like a water balloon. You can mutate it a bit.
Fluid mosaic. Mosaic.
Again, here's the structure of phospholipids in case you. Okay, so movement across the plasma membrane. Few things can move by themselves across. So water is
Polar.
what is its chemical formula?
H2O.
H2O, carbon dioxide?
CO2.
CO2? Okay, ammonia I can't remember, but does somebody have done it before? It's NH3 or NH4?
NH4.
It's one or the other. And then oxygen is O2. Okay, so these are small molecules. There's only a few atoms in these. Okay, these molecules that are very small can go across the membrane. But other molecules need carrier proteins. These are the proteins embedded, or what's the other word for embedded?
Integral.
These are proteins that are integral in the lipid bilayer. And again, the fluid mosaic model describes this fluid nature of the lipid bilayer with the proteins in it. These in A are the molecules that can just go through it easily because they are so small.
Protein channel.
Okay, these can go by themselves. Then there's another group called lipid soluble substances. What is soluble? Can dissolve into what? Lipids. It can dissolve through the lipid bilayer to get through. So often hydrocarbons. And then water soluble substances cannot go through. Why can they not go through?
They're bigger than the hole. They can't.
But what is the middle part of the lipid bilayer is hydrophobic. So if this tried to push through a little bit, there would be resistance and it would push it back out. And then ions, why can they not go through?
They're too big.
Why can they not go through?
Because
They're actually very small. They're smaller than these.
they are positive.
Huh? They have charges. They are positive or negative. So that makes it hard to go through this polar, non-polar region of the lipid bilayer. Okay.
Can they transfer through
They can go through the channels, but right now we're just talking about the lipid bilayer. Notice there's no proteins in this picture going through, so there's no transporting proteins there. So membrane proteins include channels or transporters. These move molecules in one direction.
One direction. You're insecure.
There's another side.
What's that red thing?
What red things? This is my little dot. Okay, so these channels or transporters, they move molecules in one direction. They have a lot of history. I was making a corny joke for you. They have a lot of history with these molecules. Then receptors recognize certain chemicals. So a receptor can only work with certain molecules going through. It can't, if it's made for glucose, it cannot take ribose. Okay? Uh-huh.
Yeah.
They have a similar function. They both move molecules in one direction.
What's the difference? Like protein channels or more like developed?
There's a size difference. Well, I think I have a picture of it here in a little bit. The receptors recognize certain chemicals. Okay. A few others. Glycoproteins identify cell type.
Yay.
So it could be a muscle cell up against another muscle cell, or it could be a muscle cell up against a blood cell. And then enzymes catalyze production of substances. So these are all the things that can be in the membrane. What was number one?
Channels and transporters.
Channels and transporters.
Yeah.
Number two? Receptors.
Yes.
And number three? Glycoproteins. And number four? So these are all... Sorry? So these are all the different kinds... These are all the different kinds of proteins that can be in the membrane. Alright, please focus. So here's the whole structure of the cell again. There's the nucleus with the rough and smooth ER. From the ER come these little tiny vesicles that go to and from the Golgi. And the Golgi has vesicles that go to the ER or to the membrane. And the membrane can make its own vesicles.
Oh yeah. So
yes, the question here was that the Golgi can make vesicles in between it and the ER, and the Golgi can also make vesicles between it and the cell membrane. Yes. This is the... Sorry?
How can they make vesicles between Golgi and ER?
So what happens in the rough ER?
Ribose?
What do the ribosomes, what do they... They synthesize proteins. What does the Golgi do?
They pack them.
They in and out.
They modify, they change the proteins. So the way that this can pinch off is what is the surrounding all of this?
Cell membrane.
A cell membrane. So it's all the same membrane. It's all basically pinching off of the outside surface at some point during its creation. So the reason that they can pinch off here and here and here and here is it's all the same membrane. Okay.
Okay.
So there's a few different types of movement. Passive transport, active transport, endocytosis, which includes phagocytosis and pinocytosis, and exocytosis. Passive transport, we discussed last chapter, requires no energy, and it moves due to a gradient from a higher concentration to a lower concentration. This can also be a higher pressure to a lower pressure, or a higher charge to a lower charge. Okay. They move to equalize the gradient. What is another way to say equalize the gradient? Equilibrium. Or isotonic. Yeah, so an equilibrium or an isotonic state. Different types of passive transport include diffusion, osmosis, and facilitated diffusion.
What is facilitated diffusion? Diffusion isn't the diffusion. Like, the diffusion is separate into two categories, the facilitated and simple.
Yeah,
What is facilitated diffusion?
because it's two different types of diffusion. Simple diffusion and facilitated.
What is facilitated?
We'll get over it in just a little bit. Diffusion, we did this before, is when a small group of molecules diffuse all over the whole entire space slowly. What did we look at in class that did this?
The jelly.
The food dye, the food coloring. The food coloring diffused through water. What quickened the process? Temperature, stirring.
Adding heat.
So temperature, adding heat. Good. Osmosis is a special form of diffusion. The fluid flows from a lower solute concentration. And it's only water.
Yay!
It can go into a cell or out of a cell.
Is this semi-permeable osmosis? So the solving is like the general whole thing, and osmosis is like semi.
Okay, so osmosis is that it requires a membrane, and I thought the picture was on here. But review of these terms. Solvent plus solute is a solution. A hypotonic environment is when the solutes in the cell are more than outside, and then the outside solvent will flow into the cell. An isotonic solution is where solutes are equal inside and outside of the cell. This is equilibrium. And hypertonic is when solutes are greater outside of the cell and fluid will flow out of the cell. Okay. And here's a blood cell specifically. If it's in its isotonic state, it looks like a normal red blood cell. If it's in a hypertonic state, what's going on in a hypertonic state?
Plasmolysis.
The solute goes out.
It shrinks.
What salt content or solute? Where is it higher, inside or outside of the cell?
Inside.
Outside of the cell. Okay? So water moves out to equalize the water on the inside and the outside because there's a high concentrate of salt or solute on the outside. So it shrinks down in size, and yes, it dies.
Substance have more concentration gradient than the outside since the flows like high to low.
No, the water is the higher concentration here, so it moves to a level of higher water to lower water compared to the solute.
Lower water to higher?
Higher water inside the cell to a lower concentration, like it's a ratio versus the solute. So the ratio of water to solute is higher here. So in the cell it's like 8:1, out here it's 1:1, so it's flowing outward. Okay, and in a hypotonic solution, it becomes swollen, and then it lyses. What is lysing? It bursts. What happens in a plant cell?
It gets turgid.
It becomes turgid. There's a point where no more water can come in. So, turgid. So, so what,
Plasmolysis is the same thing as lysing.
I go on the hypertonic.
it's,
Plasmolysis is a shrink thing.
isn't it, yeah, plasmolysis is the inside. Let me look real quick. Let me pause this. Another word for this is plasmolysis over here. Plasmolysis. Okay. Facilitated diffusion is differentially permeable membrane. So they use channels that are specific, and they help molecules or ions enter or leave the cell. Channels usually are transport proteins.
Oh, so they have a specific type of thing they attach.
But the thing that makes it facilitated in diffusion, what is diffusion? What kind of transport is it?
Passive.
Passive. And what does that mean?
No energy.
No energy is used. So it's still going along a gradient. So no energy is used, even though there's proteins there. So differentially means choosy, in a sense.
Arrogant.
And here's the process of facilitated transport. The protein binds with the molecule at the binding site, and the shape of the protein changes. Notice how there's kind of a double curve, kind of like a violin or something, and then the shape changes. Alright, the shape changes, and then the molecule moves into the membrane.
Isn't that the same thing as endo? Like,
Very similar, but this does not use ATP. So this does not use energy. Okay. In the active transport, molecular movement requires energy. So that means it's against a gradient. In passive transport, it's with the gradient. That is why there's no energy necessary. So active transport is often against the gradient or something really big.
Concentration and
Okay, so an example of this is the sodium potassium pump. Outside of the cell is the potassium, and inside the cell is the sodium. Okay, that's the ATP there, yeah. So they want it to get through. In order for this to happen, the ATP has to attach to the protein to allow for this process to occur. So first the sodium can go in, and then it kind of unlocks. So it goes from like this to that, and the sodium can release, and the potassium can come in, and then it closes back up, and the ATP releases.
What if 4 sodium goes into the channel? Are they going to produce more potassium if they?
So if 4,
like there is a specific number between
it's 3 to 2 technically.
What if 4 sodium enters the channel?
3 to 2.
Like more than 3 can be possible.
So it would be multiples probably.
like 6 to.
It's a constant sort of thing going on. So so it's always just 3 to 2. It's back and forth.
Isn't it always like the ratio?
Oh.
Yeah, but it's just a pretty constant unless it's not necessary.
Is sodium 3 or potassium?
Sodium.
Sodium 3, potassium 2.
So the upper region is more positive. More.
The upper region? No, it's the ATP controlling this gate.
No, so it has more positive charges for the outside of the membrane.
With these questions, I'll post a little bit more about it in the video. Okay. I'm continuing this without the class. Endocytosis is the movement of large material. So this is large molecules or macromolecules, organisms. This could be little bacteria or something that the cell's eating, and particles. And this can be things that the cell doesn't want, kind of dirt, or things that it wants to break down to use for raw material. And this movement is into the cells. Into the cell. Into. There's types of endocytosis, which is bulk phase, which is not specific, and then receptor mediated, which is specific endocytosis. The process of endocytosis is when a cell contacts the substance that it wants to transport, either an organism or macromolecules or particles, and the plasma membrane folds inward until it completely surrounds the substance. Once the substance is completely surrounded, the membrane is pinched off inside the cell to form a vesicle. Okay? This can happen in phagocytosis, which is the bulk mold, This happens with bulk material or organisms. And then there's penocytosis, which is the movement of bulk fluids or solutes across the membrane. Again, this phagocytosis is cell eating, penocytosis is cell drinking. Exocytosis is the reverse of endocytosis. This is exiting the cell. So the cell discharges material. It is the process whereby vesicles in the cytoplasm fuse with the plasma membrane and release the contents of their solution outside of the cell. Cells use exocytosis to secrete materials produced by the cell, such as hormones or proteins, or to release waste from food. Again, the vesicle moves to the surface, the membrane fuses to the vesicle, and materials are expelled outside of the cell. Here is an actual image of this happening.